Okay, well, hello everyone. Good morning, good afternoon, good evening. I guess where you're based around the world. Today we've got a really exciting talk from Tom and Linglei, discussing pharmacokinetics and biodistribution of therapeutic extracellular vesicles. This is going to be a very good talk, a very interesting topic. If anyone's got any questions, please just put them in the chat, and I will go through them. I will then ask you to unmute and ask them at the end of the talk, and I'll try and group them. If we don't get around to everyone, the transcript will be available to the speakers and they will answer them and get back to you at the end. But without that, thank you to Ken for hosting this, and over to you, Tom and Linglei.
Thank you, Will, for your introduction. So today we will talk about pharmacokinetics and biodistribution of therapeutic EVs in mice and in macaques. So that's basically the postdoc project that I've been working on in Ken's lab last year. And Linglei was also involved in this project, especially very recently in collecting more recent data. So we're presenting today. I'll start and she'll take over halfway. I'll probably don't have to convince you about the therapeutic potential of EVs. I think you all know that EVs can be loaded with various therapeutic molecules to target a wide range of diseases and molecules that you can fit into EVs. You can think about small chemical compounds, but also small interfering RNAs, short hairpin RNAs, CRISPR-Cas9 complexes, but also therapeutic proteins. And on top of that, EVs can be engineered to target specific tissues via selective retention. And there are even some reports that EVs can cross the blood-brain barrier, making them very attractive drug carriers to treat neurological diseases. I've looked into the literature and I came across this systematic review in which they summarized 157 publications on preclinical uses of EVs. And from this, it is very clear that to date, most animal models used for these studies are mouse. A very small minority of studies used rats, and only a few use zebrafish. And on the graph on the right, you can really see the trends over the past years. Mice research has taken a big jump, slowly followed by rats. But a disadvantage of using mice is that they're genetically quite distinct from humans. So we may need to resort to larger animal models in order to translate these findings further to humans. Furthermore, the administration routes are really important when you want to evaluate the biodistribution of EVs. And these are some figures from that same systematic review. What you can appreciate is that the far majority of studies have administered EVs via intravenous administration, followed by local administration, intraperitoneal, and a very small set of studies tried different routes. And again, on the right, you can see the trends over the past years. But there are only a handful of studies that really compared different administration routes in parallel using the same EVs in the same animal. Here I show you two examples. One is a study from Wicklander et al. Published in the journal of Extracellular Vesicles a couple of years ago, where they compared intraperitoneal, subcutaneous, and intravenously administered EVs from 293T cells. And you can appreciate that there are slight differences in the organ biodistribution. And another paper I will briefly show is the one on the right from Betzer et al., in which they compared intravenous versus intranasal administration of gold nanoparticle labeled EVs from MSCs. One of the most striking conclusions they draw here is that uptake in the brain was more efficient via intranasal administration, both after 1 hour and after 24 hours. So that really encouraged us to also look at this, this comparison of administration routes. But there are also many unresolved questions on the pharmacokinetics of EVs. One I briefly addressed already, like what is the best administration route, but we also don't really know what doses are appropriate and whether the dose and the administration route affect the fate of these EVs. Another question that's really important for cancer treatments, for example, is can we repeatedly administer EVs? So we combined all of these research questions. So we have compared whether intravenous or intranasal administration of EVs affects the biodistribution. And we also looked at the EV dose and tested whether this affected the circulation time and the distribution over different biofluids. To do this, we made use of a non-human primate model, the pigtailed macaque. As an advantage over smaller animal models is it is biologically closer to humans. It is possible to sample larger volumes. Also, repeated sampling is possible, which allows the tracking of the clearance of EVs from a single animal. But as a disadvantage, the detection of EVs is challenging. And to illustrate this, I will resort to kind of a cliche, the needle in the haystack. It is really like that. So if you think of a mouse of a haystack that is about this size, I think it's already pretty difficult to find your needle, but macaques are many times bigger, so it becomes a bigger challenge. So in order to get good data from a model like this, sensitive reporters are key. And one of the reporters that we used for highly sensitive in vivo tracking of EVs is named palm-RED. This is a palmitoylated EGFP nanoluciferase fusion protein that via this fatty acid group becomes incorporated into the EV membranes. This has been designed by Charles Lai at the University in Taiwan. And the nice part about this protein, it not only has GFP, but it also carries a nanoluciferase. So this is an improved version of a regular luciferase, but it is about 150 times brighter. And also because its detection relies on an enzymatic reaction, you have a very high signal over low background. Well, we didn't want to count on just one reporter. So as a second reporter, we chose the recently developed Menglo 700 nanometer dye, which is a near infrared self-quenching membrane dye. Many dyes that are used for EVs, they give a lot of black background fluorescence, and they may form micelles. With this dye, that is a lot less. So when it forms micelles, it quenches itself because of the high density of dye molecules, but only when it associates into a lipid membrane, the fluorescence is turned on. And this label has been used previously to study tumor EVs in zebrafish. And another nice feature of this dye is that at this long wavelength, there is less autofluorescence, which is good for imaging purposes. We produced palm-RED and Menglo containing EVs in XP293F suspension cells. So this is a HEC-derived cell line, which gives a really high EV yield per milliliter of culture medium because they're grown in suspension in shaker flasks. This also makes them really easy to scale up. And in literature, you can find that they're increasingly used to produce therapeutic EVs. So we produced 6 batches of conditioned supernatant, 700 mLs each per via transient transfection. Here below, you can see the pictures of the transfected cells. They're nice and green. And then we purified the EVs. First, we depleted the cell and the debris via low-speed centrifugations, followed by a filtration step. Then we concentrated the EVs via TFF. We labeled them at that stage with the Memglow dye. And we concentrated them further on an Amicon spin filter, followed by size exclusion chromatography on a QEV10 column. So these have a very large volume. The EV fractions we concentrated further, and then we stored them at -80 degrees. So I'll quickly go over the characterization. On TEM, you can appreciate that structures, the shape of EVs are found in the early fractions, whereas the protein fractions are mostly devoid of EVs. On Western blot, all the EV markers are present in the first 4 fractions, and calnexin is not detectable there. So we have EVs of a reasonable purity. I also performed a nanoluciferase assay on these different size exclusion fractions. So we find the nanoluc reporter back in the EV fractions, but also in the later ones, which really shows that it is important to separate your EVs from all sorts of protein contaminants. One other thing we did to show that our reporter is enclosed within the EV membrane is protease resistance test. So we subjected these EVs to protease K or protease K and Triton treatment. And what you can appreciate is that the nanoluciferase signal is retained upon incubation with protease K only, but is lost when the EV membranes are lysed with Triton, showing that our reporter is protease resistant. We also use the ExoView platform to see in what EV subtype our reporter localizes to. We found more or less equal distribution of the reporter over different tetraspanin containing EVs. Maybe the CD63 EVs contain a little bit more of the reporter, but you can see there's a bit of variability in these repeated measurements. So we can say that our reporter labels all major EV subtypes. We recently took it one step further. We performed a max-plex characterization to figure out what other surface proteins are present on our EVs. So for the people who don't know this technique, you can read all about it in this paper. It comes down to the following. This is a kit with a pool of 37 beads, which differ in FITC and PE signal. So you can separate all of these populations by flow cytometry. And each population of beads is carrying a specific capture antibody. So you can capture each EV subtype on each separate bead population, and then stain all of your EVs with anti-CD9, CD81, and CD63 coupled to APC. And then you can run it on a flow cytometer to assess what other markers are there. So besides CD81, CD63, and CD9, we also detected CD29, which is integrin beta, CD326, which is EpCAM, and let's see, my thing is in the way. Oh yeah, CD146 called MCAM, among a couple of others. So now we have a better idea of what markers are there as well. Another thing we tested before we went in vivo is how sensitive can we detect these EVs in plasma of healthy macaques. So we did a spike-in experiment followed by serial dilutions under 100,000 fold range, which you can appreciate here for different batches of EVs. The detection is really linear, and we could detect EVs over this entire 5 log dilution series. giving us an estimated limit of detection of about 20 EVs per microliter. So that was pretty good, I think. So now I will describe our in vivo study setup. We have performed a dose escalation study in intravenous and intranasal administration to begin with in one animal each. So what we do is we inject the EVs, we collect blood and cerebrospinal fluid at different time points after administration, up to 24 hours later. And as doses, we started relatively low with 7 times 10 to the 10 EVs, up to 9 times 10 to the 12. And each subsequent dose is 5-fold higher than the previous one. So how do the data look? I will first show the data from intranasal administration. So what you can appreciate here, after intranasal administration for all of these doses, we couldn't detect any EVs in the cerebrospinal fluid. And in plasma, it was more or less the same. Even at the highest dose, there was maybe a very small bump, but it was very close to the detection limit. So this was a bit of a surprise, maybe also a disappointment that we couldn't detect any EVs in these biofluids after intranasal administration. But we'll get back to that. How about intravenous? That looked a bit better. So for the highest dose and the one second highest, we could detect EVs in the cerebrospinal fluid after 30 to 60 minutes. And so this shows that EVs can go from plasma into the CSF and maybe from there also could penetrate the brain perhaps. But for intravenous, and the clearance in plasma, we also looked at that. So what you can appreciate is that with the increasing doses indicated in blue, the signal in plasma got higher and higher until we reached a dose number 4 in pink. That was a bit remarkable. We didn't see as much signal as with the second highest dose, and the signal decayed a lot faster than we expected. And I went back, I rechecked my particle concentrations of that 4th dose. I rechecked the nanoluciferase signal, and all of that was okay. So that gave me the idea that we were looking at a biological phenomenon. Yeah, here I show the half-life of these EVs in relation to the dose. So what you can see for the lowest 3 doses, half-life was around 40 minutes, but for the highest dose, we went down to about 10 minutes, which was really surprising. Then we wondered, what is causing this accelerated clearance of EVs from plasma? Do we see this effect because the last EV dose was way too high? Or do we see that because we repeatedly administered these EVs? So recently we got some new data. This is the first time I'm showing this. So we took a new naive macaque and we changed the order of administrations, starting with the highest dose, then going down to the lowest one, and then slowly increasing it over time. And surprisingly, we sort of saw the same pattern. So for the 4th dose, the signal was extremely high, but the clearance was relatively slow. For the other doses, the clearance was comparable, as you can see in the graph on the right. But after the 4th administration, we were again down to about 10 minutes until the EVs were cleared. So this is an indication that perhaps the repeated administration is key and not so much the dose per se. So now Linglei is going to take over.
Yeah, and then we said, so for this very interesting accelerated clearance, we want to further look into why, what leads to this accelerated clearance. So first, first we check the different inflammatory cytokine. So we only found upregulation of IP10 and IL-8 and MCP1, but we didn't find any major inflammatory cytokine expression, such as the IL-10, IL-1B, and IL-23, and interferon gamma, and also TNF-alpha. Those major inflammatory cytokine, we didn't see the strong upregulation. So those slightly, we saw this slightly upregulation of MCP1 and IP10 and also IL-8. This result is consistent with the previous report in mice. And then we are trying to, so this looks like those data leads to the conclusion that this inflammatory response are unlikely, and the explanation of those accelerated clearance we observed in macaque model. And then we are keep looking and researching why this will be possible. And we learn a lesson from a liposome field at the same time. Here, I want to share with you a study of the effect of repeated injection of pegylated liposome on their pharmacokinetics in rats with you. So in this case, the rats have received either one dose of radio-labeled pegylated liposome or 2 doses. And from the time course profile we show here, you can clearly see that the second dose can rapidly clear from the circulation when compared with the rats who only receive the single administration, especially the rats who receive the second dose at day 3, day 5, and day 7 post injection shows a faster clearance. And the 5 days post injection give us the high shows the highest clearance rate here. And then the author starts questioning why this leads to the accelerated clearance in the rat model. Can that be the effect of cellular immunity? So then the histological slice of liver and spleen were collected from animals that which have received only 1 dose of liposome and also repeated liposomes. So, and then there's no significant histopathological changes were observed between those groups. And also the author further counts the density of a sinusoidal walls, sinusoidal walls cells density in the liver and the spleen. And again, they didn't find the significant difference among these repeated injected groups and the control groups. This indicates that the first pegylated liposome injection did not lead to the increase of Kupffer cells in liver. So it can be the effect of the hepatic clearance and the splenic clearance. And then from the left figure, the author shows that indeed, after the first liposome administration in the repeated experiment, you can see that the liver, so the liposome can accumulate it, the accumulation of the liposome can increase in the liver and the spleen, especially at day 3 and day 5 post the first dose administration. When you look at the right figure, the authors calculate the hepatic clearance, splenic clearance, and then they see a very significant increase in the hepatic clearance. However, less splenic clearance rate was less increase in the splenic clearance rate was observed. So then, can it be And so then the third possibility will be candidate because of the humoral factors. And to further solve these questions, and the authors conducted a serum transfusion experiment. In this, and then healthy rats were transfused with the serum from either non-treated healthy rats, or they are treated with the liposome treated serum, or they are either in this case, like the rats are either received the serum from non-treated non-liposome treated serum, or from the liposome pre-treated serum. And from these results shown here, you can see there's a very significant decrease in the blood concentration, and also a very pronounced increase in the hepatic accumulation. And then the complement system is known as one of the major serum opsonins that enhance the clearance of liposome from the blood circulation. So then the authors further conducted a CH15 blood test, which is a high screening assay for the activation of the classical complement pathway. And then they found out that the second dose did not cause any significant consumption of the complement system. Taken together, so it looks like the accelerated clearance of liposome in the rat model are contributed both by the cellular immunity and also humoral immunity. So this will be the major explanation for this accelerated clearance phenomenon. So we think this is a good example, and it may also very likely to explain our observation in this macaque model. So after the uptake, after the very high signal of high EV associated signal was observed in the intranasal group, we further checked the immune cells, and as the circulating white blood cells contribute also to the clearance of EVs from blood. So we start looking at the PBMC. So the PBMC was isolated from the macaque from both intranasal group and also intravenous group. And in the intranasal group, and then there is no significant EV-associated luciferase signal can be detected at the various 4 doses. But this is not a case for the intravenous group. In the intravenous group, we can clearly see the very high signal, and even after 1 minute post EV administration. And also we did the calculation. We found out that the EV uptake is in correlation to the EV dose we administered. So the higher dose we give to the macaque, then the higher dose we can trace back in those PBMC cells. And then we next saw to it would be very interesting to determine what will be the PBMC subsets responsible for this rapid uptake of EVs in blood. Well, so we did so next slide. Yes, thank you. So we observed the EV uptake in the monocytes, and also we observed the uptake in the CD3 positive T cells, and also we see the uptake in the CD20 positive B cells. And also from the results, you can see that the uptake in those 3 types of immune cells occurred even 1 minute post administration, and it reaches the peak at 3 minutes. And also very interesting that we calculate the uptake efficiency, and we found out the uptake, the maximum uptake rate for the monocyte is 13.8% at the time point of 3 minutes, and for the T cells it's 6.8%, and notably, and then we see a very high uptake efficiency in the CD20 positive B cells. So this is a very, very interesting phenomenon we observed in this project. We then also investigate how those intranasal and intravenous administration routes can affect this biodistribution of our labeled EV in different organs. So first, we also compare with the mice model. In this study, we administer the EV, labeled EV, to the mice, and we perform in vivo bioluminescent imaging. And post-administration, between 10 to 20 minutes, we can see that from this figure, we can see that the intranasal EVs are remain either in the nose or in some of the mice, like those EV signals are rapidly accumulated in the liver, and in the intravenous group, and after 16 minutes, we can see majority of the EV signal are accumulated in the liver. And then we next perform the necropsy, and then we took out the organs. We perform ex vivo imaging, and we found out that the majority of the EV associated signal can be found in the lungs in those intranasally administered group, and EV associated signal can be found in the liver in this intravenous group. And we also study the EV signal in this tissue homogenate in these major organs. And so from the results we show here, we can see that in the intravenous group, the the liver spleen and then shows the highest signal, and yeah, we can trace back the highest signal in these two organs. Slightly signal, we can also detect it in kidney and also colon and brain. And however, in the intranasal group, we are only able to see a bit high signal in the lungs, but also this data have a very large variability here. We next check at the tissue homogenate from the macaque. When the highest dose was administered in the intravenous group, we see that the majority of the EV signal can be detected in the liver and the spleen, but not in other major organs. And in the intranasal administration group, we can hardly trace any EV associated signal here. So we then question where does those EV goes in this intranasal group, and we then perform a nasal flush experiment. So after the EV was administered intranasally, we collected different biofluids, including plasma, CSF, and we also take this nasal flush for detection. And surprisingly, and also it makes sense that we found out that a very high nanoluciferase signal can be detected in the nasal flush. So confirming that the EV signals, actually the EVs are remain in this nasal cavity. So our findings leads to the conclusion that firstly, the highly sensitive nanoluciferase reporter can be a very useful tool to study EV biodistribution in macaque model. And second, the circulation time of EVs in macaque model, especially in the plasma, is longer than in mice model. And third, the very interesting finding what we have here is the repeated administration to macaque model may also lead to the more rapid clearance, similar as people see in the liposome field, that the rapid clearance can be observed in the rat model. And then we also conclude that intravenous administration can lead to the uptake of EV in the PBMC and predominantly by the B cell type subtypes. And all our study or our results also suggest that the administration route can strongly affect the pharmacokinetics and the tissue distribution. So yeah, and then when the intranasal EVs will being retained in the lungs, and then it looks like then the intravenous group can have a better bioavailability when we're considering the therapeutic use of EVs in clinics. We also would like to go to the acknowledgement. We want to thank our colleagues in Vitva Lab, Lyle and Marta and Lilia, and also the rest of the group members. We acknowledge our veterinarian team colleagues, Beth, Erin, and Suzanne. And also we want to thank the our imaging in vivo imaging collaborators, Jiang and Guangshu. And we thank Charles Lai for providing this very helpful tool for the field, and all the co-authors who read our who give our suggestions are acknowledged here. So thank you for your attention.
Thank you very much, Tom and Linglei. That was a beautiful talk, as has been said in the comments. That was a wonderful talk, very interesting and very powerful data that you've generated, and very useful sort of to listen into. We have got quite a few questions, and I'd encourage everyone to keep asking these questions. So the first one was regarding, his question is regarding the purity of the sample. Transmission electron microscopy of the EV fraction in figure 1b shows a large amount of non-EV debris. Further, in figure 1d, you showed that PalmGret does label non-EV components of cultured media. How do you think this could impact the observed biodistribution? And have you got any current or future efforts to address this?
So I think that's one for me. Indeed, there were some non-EV proteins in the EV fraction, and I'm not entirely sure how that is possible. But I mean, we purified the EVs mostly by concentration-based methods. So the whole EV sample becomes a lot concentrated, and then you try to separate it out via size exclusion. So perhaps some proteins may stick to the outside of your EVs, causing the things that we saw on TEM. In the preprint, we also reported the protein particle concentrations are not extremely pure, but well, we separated EVs and proteins to a large degree. Regarding that free nanoluciferase reporter, I think the stuff that we see in the later fractions having nanoluciferase activity, that's probably because of cells that have disrupted during the culturing process. Maybe a little bit of a reporter may get secreted as well, but that's what we separate out during size exclusion. Hope that answers your question.
Yeah, great, thank you. I'm happy to, I have a few thoughts on what might have caused that debris to get through the SEC and the TFF, but happy to share that offline. Thanks again.
Yes, my question, I had two questions. So with the Western blot, I was wondering if you did any other protein like contamination checks, or if you used other markers. Another question was specifically on table 1, because I found it interesting that the EV protein concentration was increasing and pretty substantially as you noted the decreased viability of the cells. So I'm wondering if you could comment on those two things.
Yes, of course. On Western blot, I just checked calnexin, as it's used a lot. But yeah, you're right. You can always think of more thorough controls. I didn't see strong calnexin bands and I checked each of these batches. So this is rigorously controlled. But yeah, you can think of more things you can do. And then regarding that supplementary table, that's from the preprint, I suppose. I didn't notice that relationship, so thanks for bringing that up. It was yeah, tricky to keep the viability high, as these cells are really meant to mass produce proteins, especially pharmacological proteins, for example. And the manufacturer's protocol even suggests you can culture these for 5 to 7 days after transfection. We only took 3 days in order not to decrease the viability by too much. So yeah, I try to keep it as high as possible, but I agree with you. It's not extremely super good as you would normally want. Maybe there's one thing I can still add to that. So it was already a challenge to produce these huge amounts of supernatant. It was tough already.
Yep, it was. Can you keep plasma or tissue levels of EVs up if you administer by chronic IV infusion rather than single injections?
Ooh, that's a good question.
What do you think?
I would think you could theoretically. I mean, if you keep feeding EVs into the bloodstream, you will be able to keep the circulating levels high enough. But yeah, our main question was like, how rapidly are they cleared, and how does that relate to the dose?
I mean, the plasma levels, I can just add, the plasma levels will depend upon the balance between delivery and clearance. Mm-hmm. And for therapeutic purposes, if you need a certain concentration to deliver an agent, it seems like chronic infusions might be an alternative to single injection strategy.
Totally makes sense. I just wonder if it's feasible to gather enough EVs to be able to administer them over a long period of time. I mean, yeah, people are looking into the scale up and the producibility of this material, but I'm not sure how feasible it is at the moment.
Then we had a question from Bohong Zhang asking if you have an estimation of the percentage of EVs that were taken up by peripheral blood monocytes via EV route.
Yeah, that's a tough one. Also, one of our reviewers asked about what percentage of the input EVs are detected in organ A, organ B, organ C. And I think that's quite a difficult one to address. I mean, we know the input, but the percentage of PBMCs that you sample, or the percentage of an organ which you turn into a homogenate, is fairly small. So how representative is that sample for everything that's floating around? So I cannot really put a percentage on it from the top of my head. And also, there are even compartments that we didn't sample. Think about the endothelial walls. Maybe some EVs get to muscles, or maybe they just fall apart in the serum. That's also an option. So that's a difficult one to address.
Yeah, a difficult question and a good answer.
Yeah, but a good one though.
Yeah. And then we had quite a good question from Quinling Fu. If you want to unmute and ask your question.
Okay, can you hear me?
Yep.
Yeah. It's very interesting work. I just want to know, did you cut the brain section to observe the EV location after RN or RV administration? Because in your paper or in your presentation, you just observed the EV signal in the brain tissues. And I observed that there was still some EV signals in the midbrain of after the RA administration, and in medulla with the RV administration. So I'm just wondering that even the low level of EVs should have some function or have some activity. Actually, in our lab, we also checked, currently we checked the location of EVs after the RN administration in the monkey using M-cherry labeled EVs. It's a RFP, and we found clear the EVs in the AUN positive neurons and RBA positive microglias in olfactory bulb and even in spinal cord. And we did not finish our work for the brain sections for the other brain, the other brains, because the brain of the monkey was very big. So I just want to know, because my question is, maybe the EVs, even the low EVs in the brain, there maybe have some function or have some activities. So we currently we are checking the behavior of the monkey after the RNA admission of EV for the monkeys, and maybe we can get the result maybe one month or several months later. And so in our lab we have the positive result after the intranasal administration of EVs. We use the spinal cord injury models. So this paper just give me negative result. So I just want to know what's your opinion about it. Thank you.
Shall I start, or do you want to take this one, Linglei?
Oh yeah, I can start. So yeah, thanks, Dr. Fu, for sharing your experience, and also I think actually it's good to hear that you can find some EV signal when you intranasal administer to the macaque in your lab. So so far, like from the data, we show that we didn't really find the signal via intranasal route in the either in the homogenate or in the tissue slice. But the readout we are using, so our project is on progress. So the readout we didn't find our EV signal is based on the membrane and GFP. But I think recently, I think maybe the nanoluciferase is maybe a more sensitive readout to check. And also I'm just so thanks for inspiring us. And also, like, when you see the positive signal in your uptake experiment, like, I'm just wondering, your dose is comparable to the dose we use here or not? Can you tell us about this?
The dosage of let me think. The dosage for the RNA is about, for the protein dosage is about let me think, is oh sorry, I can't remember. But we decided the dosage from the rat about I think 100 times compared to rat, but I can't remember clearly. But we used the brain section, we cut the section and did some RHC or RF, and using some antibody to neurons, microglias, and we found the clear EVs in these cells. Yeah, but I just think the low dosage of EVs in the big monkey brain, maybe a signal, the EV signal is very low. Maybe did you use the flow cytometry to check the signals, EV signals? The flow cytometry?
In brain?
Yeah, I just want to know the technique for your check the EV signals in the brain hominid. Flow cytometry or some other technique.
We made a tissue lysate and then looked for nanoluciferase activity in that.
Yeah, because I think the monkey brain is very big, right? And the dosage of EV only about some billion or 100 billion or 1,000 billion. It's very low for compared to the big animal brain. So maybe the signal very low and cannot determine, cannot get the strong signal. So
no, that's
yeah.
true. And I tried to do some imaging on macaque brains in the infrared channel, and even there I found a bit of autofluorescence. So that was really inconclusive, I think. One more question. So what cell type are you using? Because the differences in brain uptake are remarkable.
You mean the EV? EV? The EV
Yes,
is
EVs from what cells do you use?
MSCs.
MSCs. Okay,
Yeah.
well, that's a big
mesenchymal stem
difference.
cell derived SEV, yes.
Okay.
Yeah, because you use the 293 T cells EV, right?
Yeah, derived.
Okay. Yeah.
Okay, we'll move this on to just another one, but I'd encourage everyone keep sending the messages in, and this is where we can make some more collaborations later on. We had a good question from Andrea. I'm just scrolling up to it about the B cells. Andrea, could you unmute? Yep.
Yeah, thank you. Congratulations, Tom and Linglei. This is really cool work. So I have actually 3 questions. Why do you think are the B cells the major uptakers of your EVs in the blood? And do you think this is a cell type specific phenomena? What I mean is, if you would use EVs derived from other cell type, would you see the same, or would you expect that other immune cell populations would do the majority of the uptake? And the third question is, I was a little bit puzzled with the results that you get from the nasal flush. Why would you have so much recovery when you do the nasal flush? Does this mean that EVs are not uptaken at all by the cells in the nasal cavity?
Yeah, maybe I can answer this one. So thanks, Andrea, thanks for your questions. And then, so for the B cell uptake, and I think this is a bonus we found in our study. And honestly, we didn't really compare other like a donor, like other parental cell type EV for this specific case. So this is a bonus we got in our study. And then the second, for the EV, the nasal flush questions. So the reason why we can trace back majority of the EV in the nasal flush after 1 hour administration is because I think the mucosal layer in the nasal area is very thick. So it is the limited factors for the, so it's a limiting factors for our EV internalization. So this is what I think. Yeah, Tom, do you want to add on this?
No, I totally agree with you. The uptake by B cells was quite a surprise. I know there's a paper from R.S. Etang and Nicole Noor-Hotham from a couple of years ago where they looked at phagocytotic activity of human white blood cells and how that changes with age. And there they find also that B cells can take up a fair amount of EVs from plasma next to monocytes, if I'm not mistaken. So yeah, there are some similarities, but also some differences. So probably the cell type differences play a role there too.
Yeah.
It would be really cool to pinpoint what protein makes it home to B cells, for example.
Exactly. Yeah, exactly.
At this point, I don't have any ideas about that.
Great, thank you.
So I'll read it all. Apologies if some of this has been asked, but I'll just read the whole message. So it's for the intranasal administration. Did you use a nebulizer for the macaque? Was the administration method the same for both of the animals? Could you detect the EV particles in the brain? And also, what was the origin of your EVs? It might have a different homing behavior than what was better shown in the MSC EVs as cited by Bertzer et al.
For the administration route, for the intranasal administration, and in the data we show here, and we didn't use a nebulizer. So in both of the mice model and the macaque model, so it's more like we train the animals and we pipetting some fluids and to their nasal area. So it's not conducted by the nebulizer. But actually, it's also something we like within our team. We also have quite some discussion about this. For this kind of macaque, like which kind of nebulizer might be maybe a good starting point for the macaque model, because the nebulizer we use in human is pretty big, and for the macaque they are animals, so they they don't really like we put some nebulizer on their face, so we need a training. So I'm not sure whether like in the audience, whether we have some experts can also provide some insight on this. And then for the detecting, and then for the question, back to the question, I think we didn't really detect the EV particles in the brain so far, and both in the homogeneous data we show and also from the ex vivo imaging data we show here. And the origin of our EV is a suspension suspension 293 cell line, which is called ESPI 293. And I think I agree with Nali that the different homing behavior will do exist between our ESPI EV and compared with the MSC EV, because the cell resource is very important for the homing behavior. I do agree here.
Oh, maybe I can add one thing about the Betzer study. There's also some technical differences. So what they do after loading their gold and nanoparticles is just spinning the EVs down at 100,000 G, but you can you know that gold particles have a much higher density than EVs. So I don't think this is a very good way to get rid of any unbound gold nanoparticles. So I'm a bit skeptical if all the gold nanoparticles they report in the brain were really EV enclosed. So there are some question marks you can place there as well, but there may also be cell type differences.
Brilliant, thank you. And I know we are very close to time, but I'm going to be cheeky and ask one more question from Sally Price as our last question for today.
Hi, thank you. Yeah, it was just a question around the brain penetration with the IV administration. It looked like your two higher doses, you saw brain penetration, but not the lower doses. Just wondering whether you think that was just sensitivity of detection. So when you're sort of administering the lower amounts, we just can't see that, or whether you actually think something more interesting, more biological maybe is happening with the higher doses.
Well, we only looked at biodistribution after the highest dose, so we didn't really compare. We didn't sacrifice the animals after each dose, so I can only say something about the highest dose.
Okay, well, thank you very much, everyone, for those questions. There's a great variety. It's quite hard to choose the questions. We had so many. So Tom and Linglei, there are more questions on the chat, but also I'd encourage people to email any questions that you have. But that was a really, really interesting talk. Fantastic data, really, really neat. So I think everyone is saying and clapping and saying a big well done to you guys for a really nice piece of work. And thank you everyone for attending. I'm sure there will be a talk coming up soon. But for me in Cambridge, it's goodbye.